Hello everyone. I would like to welcome you to today's webinar entitled Using Genomics, Wearables, and Big Data to Manage Health and Disease. My name is Annette Cruz and I am the Program Manager of the Stanford Genetics and Genomics Certificate Program at the Stanford Center for Professional Development. This webinar is brought to you by the Stanford Center for Professional Development and the Stanford Genetics and Genomics Certificate Program. Take online courses in genetics and genomics and gain a greater understanding of biology, human health, and personalized medicine. Tap into the world-class research of Stanford faculty and industry experts to acquire the skills and knowledge you need to succeed in the rapidly evolving genetics industry. Visit geneticscertificate.stanford.edu to learn more. Before we begin, I would like to introduce you to today's featured presenter, Dr. Michael Snyder. Dr. Snyder is a Stanford Ascherman Professor and Chair of Genetics and the Director of the Center of Genomics and Personalized Medicine. Dr. Snyder received his Ph.D. training at the California Institute of Technology and carried out postdoctoral training at Stanford University. He is a leader in the field of functional genomics and proteomics and one of the major participants of the ENCODE project. His laboratory study was the first to perform a large-scale functional genomics project in any organism and has launched many technologies in genomics and proteomics that have been used for characterizing genomes, proteomes, and regulatory networks. I would like to turn it over to Mike now.
Great. Well, thanks very much for having me on this program. I look forward to telling you about some of the things we're up to using data to help manage people's health and ideally help manage their and ideally cure their disease. So this is an area I think many of you appreciate is incredibly hot these days and very topical, and I think one of the events that really launched this was when Obama announced that we're going to have a major initiative in precision medicine. This was in his inaugural address in January 2015, and he pointed to this DNA helix as a way in which we're going to be able to use genomics and information to be able to better manage people's health. Well, I would argue this goes well beyond just genomics. I think we're going to see an explosion of all kinds of data that will help manage people's health and predict medicines they should use at a level that's never been done before. Now, before we dive into this, let me first show you a little bit about what frames our thinking, which is that your health, if you will, it's a state. And most of the time we're healthy and in a healthy state, but there are times in which we get disease and we become unhealthy when you get a viral infection or perhaps even other nastier diseases. And these states, they're influenced by many different things. They're influenced by your DNA, your so-called genome, which is a collection of all your DNA molecules. And also the various things you're exposed to, the so-called exposomes. That would be the food you eat, pathogens you might be exposed to, various life stresses, exercise. We all know intuitively that these will contribute to your health. And I would argue a goal of us collectively is to try and understand in quantitative terms exactly how people, when they're born with a certain DNA sequence, when they're exposed to different molecules, what kind of health outcome we might predict. And I think this will occur in some, if you will, probabilistic fashion, some means in which we would be able to predict whether someone might be healthy or get disease. And just as an example, if you're at risk for Parkinson's, you probably don't want to be a pesticide worker because that makes you at higher risk for getting Parkinson's. So, and there are DNA variants that lead to Parkinson's. So again, I think this is where we're ultimately going to head. I do believe we're in a world where soon people will be getting their genome sequenced before they're born, and that will actually make it all the more important we understand what kinds of exposures lead to what kind of health outcomes. Now, I think many of you know it's possible to sequence people's DNA. So there's been a revolution in DNA sequencing technology, and it's at the point where we can now sequence someone's DNA for less than $2,000, although to interpret that DNA sequence costs a lot more. In fact, it can cost as much as $15,000 or $20,000. But that's not the only revolution in technology that's occurring. There's other revolutions in advanced technologies, mass spectrometry, the sort of instruments that you see in airports, There's very advanced instruments that let us measure tens of thousands of molecules inside of us, again, at a level that's never been done before. So we can make measurements of unprecedented scale and be able to better define what people's biochemical states are. So a number of years ago, we launched a very large project, which we call personal omics profiling, where the idea is to make very detailed measurements of people. So we make literally billions of measurements of people every time we sample them. So we analyze their DNA, that's their genome, as I mentioned before. But we also analyze all these other features as well. So something called the epigenome, which is your modified DNA, which we know is influenced by the environment and all those other things I mentioned earlier. We also measure all your RNA molecules. This is called your transcriptome. All your proteins, that's your proteome. Special set of proteins called cytokines. They modulate your immune system. And we follow all your metabolites and small molecules as well. There are as many as we can measure. We follow your antibody responses. You may have heard about this new area called microbiome. This is an area where we actually have many, many cells that live in and on us that are really microbes that are actually very beneficial for our health. They digest our food, and we study that as well. So we actually measure, most of these we measure out of blood, but some we measure out of urine. And the microbiome we actually measure, believe it or not, out of your stool sample. And so we make measurements of all these different things. Again, billions of measurements every time we sample people. And the idea is actually to sample people over time to see what keeps them healthy and what makes them, what goes wrong when they get sick. And we do standard medical tests over here as well. And we also have questionnaires. And I'm going to get back to these wearable sensors at the end of the talk today. So basically, why are we doing all this? Well, we're trying to understand why people are healthy, what keeps them healthy, and by understanding the biochemical measures of health. We also want to happens when they do get sick, and when you do get a disease, what kinds of biochemical events change during that time, and then how can we better manage that? We want to understand how these different, I mentioned omes, so the proteome is your collection of proteins, and the metabolome is your collection of metabolites, and your microbiome is your collection of microbes. We want to understand how they relate to one another. We know intuitively that the food you eat in combination with your genome and your biochemical marks on your DNA somehow affect your health, but we don't really understand how those rules work. And we'd like to understand how they work so that we'd be able to better manage your health. That is to say, certain foods can make you better by eating them, and they will be specific for some individuals. We'd like people to eat those foods and not foods that make you unhealthy. And this can be very personalized. We're also trying to understand how people react similarly and how they react differently when you're faced with, say, adverse conditions. If you have a virus that comes along, most people respond somewhat similarly in the sense that they'll mount an immune response, but there'll be personal aspects of this as well. Some people will make certain cytokines and be able to better fend off that virus, and others not so good, and we'd like to understand why that's the case. And perhaps most importantly, one major reason we're doing this project is we're trying to identify factors that can basically help manage the health of an individual. We can learn things about people that we hope will keep them healthy, and you'll see evidence of that as we go along. So basically, many of you may have known this already, but we actually started this project by studying meat. Looks like we lost some information here, but basically, we've been profiling me for now over 6.5 years, meaning we've taken over 200 samples from 200 time points from me, many samples each time point. And during this time, I've been through 10 viral infections. I've also had Lyme disease, and we basically have, as I say, been analyzing me and following me very closely over this period. And we did this because we wanted to see, again, what happens when I'm healthy and what goes wrong when I get sick. So every one of these boxes is a viral infection. There's supposed to be a timeline here that somehow got lost. But basically, during every one of these viral infections, I have gotten sick. And we take many, many samples. So for example, these are all common colds I've had. This is a rhinovirus, HRV. This is respiratory syncytial virus. That's a common cold in kids. adenovirus is another one. And so we do dense sampling when I get a viral infection, and then we typically give blood every 2 to 3 months when I'm healthy. And lately I've been trying different dietary supplements, so we've been sampling a lot. Each of these is a day number, each of these numbers. And so again, we're following what happens as I go through these various changes. And first thing we did was actually sequence my DNA. I'll go through this somewhat quickly because I'd like to talk about some new exciting stuff. But from my DNA sequence, we could predict my risk for many different diseases. And most of this I knew from my family history or what I knew about myself already, meaning I was at low risk for obesity. Nobody in my family's ever been overweight. I was at high risk for what's called dyslipidemia and high cholesterol. That shows up here as high triglycerides. I was at high risk for those. I knew that already because I knew my cholesterol was high, and I do take statins to manage that. High risk for coronary artery disease, that was known as well. One of the big surprises for my genome, though, was I was at high risk for type 2 diabetes. Basically, I went from having a 27% chance of having this in my lifetime to a 46% chance, so huge increase. And we actually were following my glucose, which is what happens when you get diabetes. Your glucose goes off. And then, in fact, that did happen to me during the course of the study. My glucose had been marching along just fine. And then it actually turns out right after this nasty respiratory syncytial virus infection, my glucose went quite off. It got very high to the point where I was, in fact, classified as diabetic. So when we first measured it, it was on its way up at 127. A week later, it was even higher. This is a better measure for steady-state glucose. It's called hemoglobin A1C. It was right on the edge. It's arbitrary, but diabetes is classified as 6.5. Hemoglobin A1C was 6.4. I was right on the edge. And then 5 weeks later, we measured me again, and sure enough, I was actually diabetic. I had crossed the threshold. I was 6.7. And that was over 5 years ago. That was April 11, 2011, because up until then, I had the world's worst diet. I ate plenty of cake, sweets, and because I didn't know I had a problem. I cut all that out. I doubled my biking, started running, and it took about 8 months, but I brought it down under control. I got my glucose back together. So this made a big splash at the time because it's the first time someone used their genome to predict their risk for a disease, and in my case, I got the disease. And in my case, it was also manageable, and I was able to bring it under control, at least initially. And it was also quite intriguing because if you notice when it occurred right after this nasty respiratory syncytial virus infection. So what we think is that genetically predisposed for type 2 diabetes, and then in conjunction with this nasty viral infection or its associated stress response, that's what's triggered the disease. Now you may think that this all has a happy ending, and it's not completely so. It turns out that some people in fact criticize and say maybe Mike, that would have gone away on its own. You don't know that. Well, we accidentally did the right control experiment, so to speak. That is to say, we kept profiling me, but we were doing so blindly. It was actually when we scaled, you'll see in a minute, we now follow 100 people just like me. And when we scaled, we had stopped looking at the data, so we were doing this blindly. And then at this point here, somebody looked at my hemoglobin A1C, which turned out to be 7.0, and said, Mike, you're at 7.0, you're diabetic again. I said, oh, I had no idea. So he sampled me. I'd stopped running, so I started running, and I could bring it a little bit under control to 5.7. These days it's actually drifted up. In fact, when I got Lyme disease, it hit 6.5, and it got up high again recently. So these days it hovers between 6.2 and 6.4. There's no question I have a metabolic disorder, and it's not fully under control because I'm right on the verge of being diabetic. So again, this is hemoglobin A1C, this is glucose, and I'm way elevated over what I should be. And it's not just me. My brothers and sisters all have elevated glucose. We're all fairly thin, so it's a little bit surprising. But it's pretty clear then that we have a genetic predisposition for type 2 diabetes, and that's what this one is, it's type 2. So basically, again, this is a case where genomics and genetics actually predicted disease risk. In my case, I could at least partially manage it. I'll probably be doing other things like taking drugs to actually get my glucose under control in the future. Now, since that time, we've actually carried out many other kinds of assays as well. We did do whole omics profiling. We really have, if you will, an IMAX movie type picture of my biochemistry as it changes during the time I get sick and when I got type 2 diabetes. But we've actually been making other measurements as well. One very fascinating area is the field of epigenetics and epigenomics. So epigenomics is a collection of your modifications to your chromosomes, if you will. And one important set of modifications is there's chemical modifications called DNA methylation. So what's interesting is that we know that your DNA can get modified in response to various environmental cues, but we don't fully understand it. It's a very fascinating field. But we know that this one in particular, modification of your DNA by DNA methylation, which is a chemical modification, it actually, we know it's affected by nutrition, it's affected by things like exercise, even your age, and other environmental cues all affect your epigenetic programming. We also know that it's associated, this modification, with turning off your genes. So we don't fully understand how this whole thing works, but we do know it's very, very important, and so the things you do really do directly affect your genes and your health. And there are now ways of mapping out all your modified DNA sites, and something called whole genome bisulfite sequencing. It gets a little sophisticated, I know, but you can actually map out all the modifications in your DNA, and there are millions of them using this technology. So we've done this for me, and it's quite fascinating. You can see very interesting regions of the genome that change. One area that we think is interesting is trying to use your DNA modifications to predict what diseases you might be at risk for. Just like we use your genome sequencing to predict what diseases you might catch, we can also look at your modified DNA to try and do that as well, to understand what diseases you might be at risk for. And this is just one example. I have a number of regions of the genome like this that are affected both genetically and epigenetically. So this is a case where I have a change in this particular gene. It's called a phosphodiesterase gene. And the point out of all this is that this gene, it turns out my father's copy is actually inactivated genetically. We all have these kinds of mutations. And in my case, my father's gene has actually been inactivated through a genetic change. And my mother's copy is actually shut off by epigenetics. So the control sequences are modified by this DNA methylation I mentioned earlier, and so we get very little activity from this gene, which we can measure by certain readouts. It's called RNA sequencing. We get very few RNAs that come from this. So the point out of all this is that this gene is inactivated two ways, one epigenetically and one genetically, and as a consequence, I actually have this, again, inactivated by two methods. And in fact, I make more eosinophils, which are involved in your immune response. I make more of these than I normally should. So we think that getting your genome sequence is not only important, but we also would argue that getting your epigenome determined as well is going to be just as important, possibly even more so, because that's something you might be able to modify through the food you eat and your exercise and such. Well, as I mentioned before, we've actually scaled this now to study 100 different people. We're following 100 people in incredible detail, just like me. We sample them every 3 months when they're healthy. And if something unfortunate happens, like a viral infection or stress responses, we'll sample them more frequently. We'll take about 5 or so extra time points to see what happens when people get sick. We also have been running people through some of these dietary perturbations, these dietary changes, to see the effect of diet on people's health. And so again, we've been doing this in incredible detail to see exactly what happens when people take supplements or certain other kinds of dietary changes. And so we've actually been doing this for now over 3 years. We've collected, this is where we were at the beginning of summer, we've collected over 1,500 time points from people. So we're really learning in incredible detail how people are when they're healthy and what happens when they do get sick. And just to give you a few examples of some of the things we learned from this, it turns out we learned important health information from almost every single person we study. So just to give you some examples from the genome sequence for the first 100 people, we've looked at 50 in detail, and 3 of them have mutations in their DNA that are very, very important for them to know about. So one individual had a mutation in this gene, which puts her at high frequency or high risk for this type of cancer called paraganglioma. And because she had her genome sequence, she'll do a simple urine test to keep an eye out for this, which she would not have done had we not sequenced her DNA. She would have no idea that she was at risk for this. Another individual has a protein C mutation, which puts him at high risk for coagulation. And a third person actually has a mutation that affects their heart rhythm and possibility of heart attack, something called, or heart failure, something called dilated cardiomyopathy. So they have a high risk for this, which got discovered partly through their family history and partly because we sequenced their DNA. And in fact, they do have some heart issues that they're taking medication for. So just from their DNA alone, for 3 people, it turned out to be very, very useful information about things they should do to keep an eye out for possible disease risk. And so, again, they will use this information to better manage their health. It turns out we're also trying to understand what happens to people when they gain and lose weight. So I think many of you, in fact, some of you may be even experiencing this when people gain too much weight and then they can lose it again. Well, what does that mean biochemically? Well, we've looked at this in our group of folks. Many of them I forgot to mention, but they're actually pre-diabetic, and there's a certain type of pre-diabetes called insulin resistance. It puts people at high risk for type 2 diabetes. Most of these folks will get type 2 diabetes. So we've been following them in detail through the same kind of profiling I mentioned. And as I say, this group of 24 people went through a weight gain, weight loss study, and half of them are high risk for type 2 diabetes. The other half are normal, healthy folks, and they're matched for their height and weight, called body mass index. They're matched for that. And so basically, we wanted to see what happens to these folks when they gain and lose weight. And what we discovered, first of all, even at baseline, even before they gain and lose weight, we can tell the pre-diabetics apart from the diabetics based on their molecular composition. So this is by looking at all their RNA molecules. It turns out the insulin-resistant folks are more elevated for certain kinds of biochemical pathways like oxidative phosphorylation. They actually have more certain platelet-specific genes that are expressed, and some other diabetes-related genes that are up in the pre-diabetics relative to the insulin-sensitive. These are the healthy folks. The same is true for metabolites. I showed you the RNA, but here's the metabolites. Turns out the insulin-resistant folks, the pre-diabetics, are more likely to have certain metabolites elevated relative to the sensitive folks, which will have other ones. This is just one example of a metabolite that's actually, it correlates very sharply with their likelihood to get type 2 diabetes. And so what's interesting about this metabolite is it actually comes from your microbiome of all things. So the point out of all this is that your molecular composition of people who are at risk for type 2 diabetes and those who aren't are very, very different. What we found, though, is as people gain and lose weight, their biochemistry does change dramatically. So these are the molecules that do change. Every row is a different molecule, and every column, this is at the beginning, this is after they gain weight, this is after they lose weight. So up here are the molecules that start out low and then they get high when people gain weight, and they go back to low. And there are other molecules that are high when they're healthy, and then when they gain weight, they actually, these molecules go down and then they return to normal. And there are other interesting ones that might start high, go low, but actually stay low. They don't quite come back. And we can actually identify these patterns systematically and see the pathways that change. And the bottom line out of all this is that when you gain weight, actually, one interesting finding was that your inflammation response goes up. So your immune response gets activated when you gain weight, and then it returns to normal after you lose weight. Another interesting pathway that changes is, it turns out, this one, your hypertrophic cardiomyopathy pathway, that's a lot of mumbo-jumbo, but what that means is that this pathway gets activated, which puts you at very high risk for heart attacks when you gain weight. And so we have a biochemical understanding now how people might get heart attacks when they gain weight. It comes from the fact that this pathway is activated when you gain weight. We've also found what turns out to be really fascinating. We found that even at baseline, even when people are healthy at baseline, their profiles are very, very different. So these are some very fascinating immune molecules called cytokines. It turns out we all have different levels of these cytokines, even at baseline when we're healthy. So this is a complicated figure, but each color is a different person. And each name here around the outside is the name of a cytokine, and this is high levels and this is low levels. And what we found, for example, is that the blue person has a lot of this one, IL-7, the light brown person has IP10, the orange person has this one, MIP1B, and so on and so forth. We all have different levels of cytokine, which probably puts us at risk for certain diseases differently. So some of us, if I were to throw a virus on a room and half the people might get sick and half might not, and that might be because we have different levels of cytokine that help us fend off those diseases. It also turns out, you've heard about your microbiome perhaps. Your microbiome is very, very important. It's the collection of microbes in and on you, and a lot of them are in your gut. And it's thought that we have 10 times as many bacteria in our gut as we do human cells in our whole body. And so the microbiome is essential for digesting your food and making essential vitamins and things like this. It's a very, very important good thing. It's not always bad like you might be worried about. But it turns out if we look at people's microbiome, everybody's microbiome is different. That's been known for some time. We all have a different microbiome, but some people can be different in interesting ways. This is two people here who eat a lot of almonds, and they're very different. This is a way of relating people to one another. In this plot, if you're similar, you'll be very close to each other. Well, these two individuals eat a lot of almonds. It's a family, a husband and wife. They eat a lot of almonds, and they're just very different from everyone else. So your diet can really shape your microbiome in long-term changes. What we found is that for short-term perturbations like the weight gain study I mentioned or viral infections, you will shift your microbiome, but you actually look closest to yourself rather than someone else. So this is just comparing people, their different time points. Every color is a different person, and it turns out that people, like this red person, if we ask who's closest to who, well, even if you gain weight or have a viral infection, your microbiome will look closest to yourself rather than someone else. So the point out of all this is we all have personal microbiomes. It shifts when we do gain weight or when we get a viral infection, and it's a short-term perturbation. It will shift, but you'll still look closest to you. Your bacteria and things are very personalized, and that's probably because we tend to eat very similar diets, and our genome and epigenome help control all this. So this is really where I think all this is going. I see a world where we'll be able to understand where people, when they're faced with different perturbations, so some people, if they eat unhealthy, they'll gain weight and become metabolically a mess. Other people, if they eat unhealthy, they actually may gain weight or they may not. But even if they gain weight, they can stay metabolically healthy. There are plenty of very obese people who don't have type 2 diabetes. And we'd like to understand biochemically why that is so we can better manage people. Certainly we can try and make sure that they eat healthy foods, but we should make sure they eat healthy foods for them. And this food can be very specific for individuals. We're also trying to understand how certain people, when they get viral infections, some get sick, but some others stay healthy. And again, we'd like to try and keep people as healthy as possible from these circumstances. This isn't the only study we're running. We're running another one where once a year we're profiling people from around the world, actually as part of a meeting that I'm involved in. It's called the Human Proteome Organization. We actually are, as part of this, we actually go around and sample people and follow their biochemical composition. We do this once a year at the annual meeting, and so we'll be running around the world taking aliquots of people to see how biochemically different they are from one another. There's other projects out there as well. I've just listed a few here. So there's one in Seattle run by Lee Hood, another in Sweden, and so on and so forth. There's a number of projects now erupting like this around the world where people are trying to understand in biochemical detail how, what it means to be healthy and what happens when people get sick. There's also a new exciting area that's going to play into this in a big way, and this is using wearables and sensors to be able to better manage people's health. So I mentioned all the biochemical measurements. Well, there's now getting to be devices that let you measure many, many different physiological sorts of things. In fact, believe it or not, there's over 500 health-related devices on the market that let you measure all kinds of different things. And I don't have time to summarize them all, but I'll tell you about a few of these. The simple ones are things like scales that you can stand on that will actually measure your weight, and they will send that information into your smartphone. There are continuous glucose monitors. This is especially useful for type 1 and type 2 diabetics. These devices actually prick onto you, and they will continuously measure your glucose. Probably the most popular devices you're aware of, these smartwatches. There's well over 50 available on the market. They measure all kinds of interesting things. They measure your activities, meaning steps, running, biking. They'll measure your skin temperature. They can measure, depends on the device, but they can measure your sleep, stress responses, and so on. They measure all kinds of things, and they're measuring, again, in a continuous fashion. And what's important is that these devices collect information, and they relay that back into your smartphone. And what that means is in the future, your smartphone is going to be your nexus of health information. So all this information is going to relay back into you, into your smartphone, and collect this. And to be honest, I use 8 of these devices every day to collect information. It totals up to about 2 million data points per day that you can collect. And even if you don't have time for all those devices, you can actually download free apps on your iPhone and get some of these as well. So in fact, this is one I use. It's a free app that lets you measure your steps, your biking, your running, and also tells you where you are all at the same time. So again, this information will all collect into your iPhone and help manage this. So why is this important? Well, if you think about it, you probably go to a doctor at most once a year if you're healthy, and they probably see you for about 15 minutes, maybe 30 minutes if you're lucky. And so out of the 370,000 waking minutes in a year, the doctor sees you for just the tiniest possible fraction of this. It's like a tiny drop in a very, very large bucket. So we think there's value to actually being able to measure people all the time. You'll collect a lot of data and a lot of information about a person over a long period, and not just in that 15 minutes where you wonder if that's representative of your health state. But by collecting this all the time, you really get a pretty good idea of what your baseline heart rate is and if it's changing and this sort of thing. I can tell you personally, what this is going to mean is that in the future, your smartphone is really going to be your command center, your control center for collecting all this information. I think ultimately the biochemical information will head in here too, and that will be able to help you better manage your health. To give you a personal experience out of this, when I got Lyme disease last summer in 2015, and my wearables were actually incredibly valuable for discovering this. In fact, because I discovered that my oxygen level was lower than it was supposed to be, my heart rate was elevated, and I knew things weren't quite right. And then I got a low-grade fever for 4 days, and in the end, I was able to figure out it was Lyme disease. I was in Norway, so it was a big deal, but they don't have much Lyme disease there. And in fact, the person recommended I get penicillin, and I told them I think I need doxycycline. And if I hadn't had the wearables, first of all, the wearables helped me find this in the first place, and then by taking command of my situation, I was actually able to get the right drug. So I think that's going to be one of many examples in the future where these wearables will feed back directly to you, and then you'll use them and go to the doctor to help better manage your health. And so we'll not only have biochemical measurements, we'll have all these physiological measurements as well. So this is Mike Snyder's future. I envision a world where people will be getting their genome sequenced before they're born. In addition to all these biochemical measures, we're going to have wearable information as well that all feeds back. Again, ideally it would feed back in real time or at least in short time. And with this, we'll be able to better predict disease risk, catch disease early, monitor disease and treat it, all based on all this information, the so-called big data. And the nice part about it is I hope in the future we'll be shifting medicine from being a reactive situation where we react when people get sick to proactive where we work to keep people healthy. That's really the goal. And so again, here's where we're at. Personal genome sequencing is here. All these other multi-omics profiling is really important. I think these longitudinal profiles are very, very valuable. Everyone is unique, and so everyone's special, not just to the mother. They're very, have different biochemical profiles. And I think we all are going to be responsible for our own health. So that's wrapping it up. I'm going to turn it back over to Annette. And yeah, and happy to take questions if people have any. I have a wonderful lab and wonderful collaborators. These are all the people who helped out in this study. So back to you, Annette.
Great. Thanks, Mike. So before we do some quick questions, I'd like to tell you a little bit about our program and our center. So the Stanford Center for Professional Development makes it possible for today's best and brightest professionals to enroll in Stanford University courses and programs while they maintain their careers. Curriculum from Stanford departments is delivered online at Stanford, at company worksites, and international locations, providing a global community of learners with flexibility and convenience and enabling them to apply their education to their work. Additionally, the Stanford Center for Professional Development has a long history of extending the world-class teaching and research of Stanford faculty to industry corporations of all sizes, from startups to mature, successful enterprises. Visit scpd.stanford.edu to learn more. So now we have a few questions. So Mike, when will this become available to everyone?
Well, right now it's a research project. So the way we carry it out, it really is just for a limited number of people. 100 folks who are following in incredible detail. But I think some form of this will come out in the future. Obviously, the wearables are there now. I think we're going to see there's starting to be some companies forming in this area about measuring a lot more data to be able to manage people's health. So I'm guessing, so some levels rolling out now. It's not covered in a typical doctor's appointment, but it wouldn't surprise me if in 5 to 10 years we'll see a lot of this activity. Certainly by 10 years, I think we'll see a lot. It won't be quite as deep as what we're doing on me and these 100 people, but I think it will still have a lot of data to be able to better manage people's health. And I didn't mention, but imaging data, some of these other things are going to be very, very important as well.
Okay, great. So with that thought, is there such a thing as too much information?
Well, from my standpoint, the answer is no. Now, some people may be overwhelmed by this, but I think you can manage the kind of information that you want to come to you. So yes, it's billions of data points, but in the end, it always distills into a few common themes, like either you're sick or you're healthy or you have a viral infection or maybe you have Lyme disease. So it won't be like you'll get flooded with billions of data points. You'll get filled with possible things that you should be on the alert for. And some people, though, they won't want this information. You don't want to wake up every morning and worry, oh, I'm going to get Lyme disease or I'm going to get cancer. That would not be a very healthy situation. So if you're a worrier and you worry about this stuff, my recommendation would be that you just limit the information that comes back to you. Only it would pop up if it seems a very strong likelihood that this will happen or there's certain simple tests you should be doing. So I think that threshold of what information you want to come back to you will differ for different people. And again, worriers should probably get less information. People who can handle lots of information, well, by all means, let them have whatever they can handle.
Yeah, great. So, I mean, with all the information and all the different devices and all of these different collection of data, what about the privacy? Who owns all of this information?
Yeah, well, at the end of the day, I think you're the owner of your own information, in my opinion. Now, you do have to be coached about this in the sense that we do have genetic counselors involved who sort of let people know that they may learn things they don't want to hear about and such, so we do want to manage that. But as far as privacy in terms of insurance companies and things, at least in the U.S., you're not allowed to be discriminated for employment or health information, although you can for long-term disability and for life insurance. So those two, you can be discriminated against, so that is a possible concern. I would still argue it's better to be know the information and stay healthy and possibly pay a little more for life insurance if you have to. That would be better than the alternative, which is to be unhealthy and not know. So it is a trade-off. Unfortunately, it'd be nice, obviously, if there wasn't any discrimination on those things. But yeah, it's somewhat limited. I haven't heard any strong cases of insurance companies discriminating or people discriminating against others based on their genetic information. Although it is, conceivable it could happen in the future. But yeah, I think, again, number one priority would be to stay healthy, and I think the information can help you do that. I think for privacy, we should try and manage it as best we can. These days, I don't think you could ever say anything is completely secure. So even if you thought you were storing it in a very, very safe place, there's a distinct chance that it will get hacked and somebody may have access to it. And that's why we'd like to avoid any discrimination based on the sort of information.
Yeah, thanks. This is very informative, and I think everyone can really take from this talk. So thank you. So again, if you like this topic and you're interested in more information, this webinar is presented by the Stanford Genetics and Genomics Certificate Program. Visit geneticscertificate.stanford.edu for more information. Thank you, everyone, for listening.
Bye, all.